Welcome everybody. It's good to have met you in person for the first time. Yeah. Before we get started, just two things for me. Sorry, I'm wearing sunglasses. I got LASIK yesterday, so sunglasses not professional, but my eyes look less so. But everything's good. Second thing, I'm going to delete the Google Calendar invite that you have from me because it's just sending people too many updates and being weird. So anyone who has attended via Zoom, it should be in your calendar through Zoom. If it's not, just go back to that Zoom link that I would have sent you in the past and that I will send you again on Monday, and you register through that. It'll send you an email confirming your registration. And in that email, you can click to add it to your calendar. So that will just save a lot of people's calendar invites. So sorry about that. And then I'm going to turn over to Mitch for announcements.
Yeah, good to be back, everybody. Got a healthy baby at home. My wife's doing great, so we're very grateful. And I'm technically back on Monday, but I'm around now, so if you need stuff on me, feel free to reach out. I don't have much in the way of announcements. Obviously, we know there's a government shutdown going on. It's probably affecting our ARS colleagues pretty severely. So another storm to weather, but we're persevering. Any other announcements? Will be good at the top.
Yesterday, Mitch joined us, and yesterday, Representative Jimmy Cleveland joined us. So Neil and I gave her a tour of the perennial flags, greenhouses, what we do, why is it important. Mostly showcasing how a one crop can produce, oil-sweetened fiber, ornamental varieties for commercial gardeners, kind of proving our work to her, if you will, of like why should forever green crops continue to be funded besides the obvious, like we need to other things can come out of the same crop, especially in the plants world. So she kind of walked away with some great talking points that she can bring to her colleagues and fight for more funding for everything that we do. It's good.
Would you mind repeating which senator or rep that was again?
House Representative Jenny Cleveland, and I believe she read.
Yeah. Thanks. She's been a long-time champ, so good to get her out there.
Yeah, just for more context. So Ginny is one of the key reasons, I guess you'd say, that we have agreed funding that covers me, Colin, and 4 of our breeders. So incredibly important, stable, long-term funding from the state. And it just so happens, so she's already a fan of Forever Green, has fought for us a lot. Just so happens that her husband, Tom, was one of Don Wise's first grad students in the early 80s. And so they've known each other for, decades and decades, 45 years or something like that. So yeah, really awesome. She reached out. She wants to put some perennial flax in her yard. It was a great opportunity for her to come visit and learn more about the state of the program today and feel some perennial flax fibers and, check out the greenhouse. So it's a lot of fun here. No. Anyone else have anything to share? I guess I'll put out there, we are, before too long, maybe in about a month, going to be releasing, or the Department of Agronomy and Plant Genetics is going to be releasing a position. Is this okay to share, Jim?
Yeah, I think so.
Yeah, so we're going to be hiring, I think it's fair to say, the world's first tenured track perennial grains breeder, which is really exciting. So that same pot of funding that I just referred to is allowing us to do that. And we're hopeful that we get an awesome person in who can really build on the program that's been built so far and lead it into the future. So that'll be coming out in about a month, and then we'll be sharing it widely and asking for people's help in getting the word out to potential candidates. Okay, that's all I've got. Take it away, Brett.
All right. There we go. Everybody can hear me? All right. So, yeah, I'm going to talk today about our project related to greenhouse studies of enhanced nitrogen fixing bacteria Pennycress. I think I'm one of the earlier presenters this semester, and I apologize because I think I was one of the very last presenters last semester. So not a whole lot has happened in terms of advancements, but this will give me a little bit more chance to kind of dig into kind of what we're doing and why we're doing it, and present a little bit more of the data we have obtained up to this point. I think it's important to first kind of go through and keep in mind what our long-term goal is with this project, and that's to improve plant growth through sustainable approaches that harness beneficial plant-associated microbes. And we have the specific goal of lowering nitrogen requirements. So that's kind of always a theme for us. We look at other things as well, but nitrogen requirements is the thing we would really like to make a dent into as far as our long-term applications. This is an agricultural systems by integrating beneficial diazotropes or nitrogen-fixing microbes. That means that we work with an enzyme that uses this unique metal cofactor. This is nitrogenase or metal cofactor called BMOCO that actually is where nitrogen fixation gets done in biology. And one of the systems that we've used for actually probably about 50 years, people have studied this genetic system because you can get nitrogenase out of this organism. This organism we work with is started with anyway is Azotobacter vinelandii. It is unique among a lot of the diazotropes in that it's able to do a lot of what we do as far as nitrogen fixation, and it's also a obligate aerobe. And that makes it very unique from what you find in most fruits, where a lot of those organisms have to be anaerobes or in some cases microaerobes. But Azotobacter actually wants oxygen, wants a lot of oxygen, and it wants it all the time. And so this is the reason people used it for years to study nitrogenase, because you could grow lots and lots of nitrogenase without having to do this under anaerobic conditions. So you can grow it into fermenters. And that's where I kind of started working with Azotobacter vinelandii. Once I got here at the University of Minnesota about 16 years ago, we started shifting our approach from studying the enzyme to really studying applications. What could we do in terms of trying to drive some of that nitrogen that this organism is able to easily grow on, getting it from the atmosphere, and maybe provide it to its neighbors. So some people told me early on that's probably a futile process because the moment you start releasing that outside, it's going to be genetically unstable. But we're stubborn, and so we pursued this anyway. So this is just kind of giving a little bit of an idea. Like I say, we've been working on this for over 16 years. This is some of the goals we've had over time. Initially we started off just saying, can we do this? Can we actually produce more nitrogen in some of these model diazotropes? And can we demonstrate that we can transfer that nitrogen to some other non-diazotrope in a way that is obvious and clear? And so, again, because of the benefit of that you can grow this organism aerobically, we wanted to consider the possibility that we could grow this with algae. And that was where when I first got hired, that was one of the things I was supposed to be looking at was biofuels, and we were working on algae at the time. And so our initial model system was based on that. And this realization as we started working in this area that if we weren't going to try to increase any type of biofuels we're producing from algae, we're going to also have to increase dramatically the nitrogen requirements. Because any biomass you have with algae is still like any other plant, it's going to need more nitrogen. And so that was the initial effort that really got us going this direction. And we went into the literature and we stole several different pieces of the puzzle from other researchers. One was this organism called Microactinium conductrix, which is an algae that at the time in the 90s, the European Space Agency was interested in this because if you're going to try to send people on long-term space flights, you've got to figure out a way to provide them sustainably with nitrogen or with carbon. And the nice thing about this algae is it'll grow in sunlight and it produces sugars. And so in a way, it's kind of a little bit like a land plant in that it's taking, using photosynthesis, and it's not just growing for itself. Most algae keep all that sugar inside, but this organism comes from a symbiotic relationship with paramecium. And so you'll find paramecium floating around with hundreds of these organisms. And it's producing the photosynthate, the sugars, in this case, predominantly maltose and glucose, and secreting that out into its environment. So in some ways, it's like a little tiny model plant, a very simple model plant, and it's producing our sugars. And then over here, we've got our azotobacter, which can handle the oxygen. That's really important for this. Is able to take in nitrogen. We were trying to convince it to produce ammonia or other nitrogen compounds that it would then share. So this was our stable system we thought of as what we were working towards. And I won't go into all the details. There's a lot of work that went into this, but around 2020, not only had we started proving we could produce ammonia, but we were producing pretty high yields. And this is showing here, we're showing the amount of sugar that presumably would be coming from the algae in this type of situation. And we show the millimoles of sugar versus the millimoles of ammonia that we're able to produce. And we can actually produce quite a bit of ammonia, this organism, where we kind of figure out how much carbon it actually takes per nitrogen. So in this case, we're getting about 1 mole of ammonia fixed for every mole of sucrose that we're consuming. And so this is our optimized system, functioning and fairly reproducible in the laboratory. So that's where we were at many years ago. And this is what this kind of looks like when it's really is working. So our wild type strains are over here on the left. These are our strains that we have enhanced to produce more nitrogen with our algae. So in algae, everything works really well. And so we kind of knocked off these first two goals of increasing that extracellular nitrogen production in this mild diazotrope, and then demonstrating that we could transfer that to a non-diazotrope. So about the time we started making progress on that, the interest in the algal field was kind of starting to wane a little bit. And we were also recognizing that the shorter term goal might be to try to look at the applications in traditional agriculture with different plant crops. And so we were looking at several different model systems. We didn't want to work with a system like soybeans. Soybeans already has a nice established symbiotic type of relationship that has developed over many millennia. So we're not looking to try to do our enhancement to an organism like that. There's plenty of people working in that field. What we were thinking is, what can we do with some of these organisms that are some of these plants that are non-nodule forming plant crops? Are there systems that we can work with, and can we just apply our systems to things like corn, wheat, some of these other crops that we thought might be a good test system? And so we started playing around with this, but oddly enough, higher land plants are somewhat more complex than green algae. Green algae don't really have the systems that fight off your bacteria if they see it as a foreign invader. And so there were some things we had to deal with to try to make this transition. And so we tried to establish what the hurdles really were early on to overcome that we needed for success with these higher land plants. And that's where this project kind of comes into play. Is we've made more progress before we got to this point, but we actually tried to jump in this project and get a little bit of preliminary data showing proof of efficacy if we went into land plants. But to do all that, we had to make some progress first on all these other fronts. And so first question we had was, can we increase nitrogen capture for bacteria that form close relationships with crop plants? And that was an issue because while azotobacter grows really nice with algae, it is not very good at growing anywhere in the presence of plants. It grows away from the plant. You find it in soils, but you do not tend to find it in close proximity to the rhizosphere. It kind of seems like it's either working with fungi potentially, or is a little bit further away. It just doesn't do very well on most plant roots. Kind of tested it. And so this is our other model that we look at when we kind of think about these, is how we really define different microbes that are associated with plants. We have 3 different classes of microbes that we're interested in. And again, most people think about the rhizosphere. We think about not just the rhizosphere, but also the actual epiphytes. That's the bacteria that are colonizing and living on the roots of the plant. And then we look at the endophytes as well. We're all very interested in the organisms that are endophytes. And endophytes is a hard definition. Some people define it as having to be inside the root itself. Other people just define an endophyte as something that is resistant to bleach. If you take out roots and you soak them in bleach for a short period of time and they're able to survive that, that's because they're somewhere in the plant root where they're protected from that bleach. So it just kind of depends on how you try to define what an endophyte is. Some people will, section out their roots and try to prove they can see the bacteria actually living inside. We use the bleach as our definition for endophyte. So we started looking at this. Again, the Azotobacter itself is not going to be anywhere near proximity in the rhizosphere, at least not from our data for plants we've studied up to date. And so we needed to identify some other diazotrophic microbes that do form close associations with different plants. That was our next real step about 5 years ago, was to kind of switch over to a different system. And these are some of the strains that are out in the literature that we thought would maybe be some good targets. There's an organism called Gluconacidobacter. There's Azospirillum, which is highly studied, Herbispirillum, which is highly studied. Klebsiella, which is another model that people are interested in, although some people worry about Klebsiella because the original name of Klebsiella was Klebsiella pneumonia, and that's because it's associated in some context with pneumonia. There's some Pseudomonas strains, and then there's a lot of other minor strains, but these are some of the bigger players that we looked at. And we did an evaluation of all these, and based on a number of different considerations, we selected another model microbe, this Gluconacidobacter diazotrophicus, which is found as an endophyte in sugarcane, and can also grow as an endophyte in many different plants. So this was only discovered, I think, in the late 80s in sugarcane. It's become quite a nice model for endophyte interactions. In Brazil, people already use this. They put it in they have strain collections and they use this as a biofertilizer. It's arguable whether that's for nitrogen fixation. All the organisms I showed on the previous slide are known nitrogen fixers. So that's why we selected these. This is known to be a plant growth promoting organism, but the reason we really liked it was there's a lot of reports that you can transfer not just to sugarcane, but it'll grow with coffee. It grows with wheat. It grows with Arabidopsis. So we had quite a few different reports saying that you could grow this as an endophyte with other crops. And all of these crops, again, are non-nodule crops. So this seemed like a really good system to use. And that's where we kind of started with. And then we attempted to apply everything that we had learned from our work for the previous 5 years in Azotobacter, trying to optimize it to produce ammonia, see if we could do similar things with Gluconacidobacter diazotrophicus. And so because we're doing that, we have to kind of go back to our starting point, say, first of all, can we actually get enhanced nitrogen production in Gluconacidobacter diazotrophicus? We're going to work with this strain. And so we kind of went back to the drawing table as far as seeing if we could apply the same procedures and same approaches to this strain. And to do that, we had to develop a whole bunch of different genetic techniques so that we could do extensive genetic modifications in these strains. We also had to learn how to grow this because Gluconacidobacter kind of sits in an odd place among microbes. It is not an obligate anaerobe, but it is not an aerobe. So you cannot fix nitrogen if you just start your cultures on a plate in the laboratory. You can do it microaerobically. So we have to grow these in an atmosphere where we give them a little bit of oxygen, about 3 to 5% oxygen. And that way they can grow and fix things diazotrophic. We also know that if you start getting enough mass and you keep the oxygen that low, you can't get your densities of your culture up very high. So once the culture density gets high enough, it does become a full aerobe. But it's a little bit more difficult to handle in the laboratory. For that reason, we build our own reactors so that we can do the testing that we do to try to accomplish the same tasks we do in Azotobacter. So a little bit harder to work with Azotobacter than Azotobacter, but we've been working on this for years and have some really nice genetic systems. It's great. And this shows the same type of approach that we have applied to Azotobacter. This is Leucanocetobacter now. These are plates where we were growing them under these microaerobic conditions with algae. This is our wild type strain with the algae. These are different modifications of the strain. And you can see some of our later strains. We're getting very, very good transfer of ammonia to our algae in these systems. That shows in the simple system with a plant that's not attacking this organism or fighting the organism, we do get nice yields of ammonia. And actually, our ammonia yields are fairly similar to what we can get in Azotobacter. So under the right conditions, it's about 2/3 as good as Azotobacter producing ammonia. So we got past that hurdle. And then we also at the same time, this was part of a USDA proposal that we did many years ago. We also wanted to try to understand the mechanisms that govern this successful plant microbe interaction. That's going to be essential for making sure that not only can we get the strain to do what we want, but we can get enough of the strain in to the plant to actually have an effect. That's a big part of where this project, where work for Evergreen is kind of come from. Is the initial stuff that we were doing here has shown, yes, we can get some Leucanocetobacter growing with some different plants. And that's where we get into some of the systems that we were using to study specifically the mechanisms that are governing this plant microbe interaction. And so this is where Pennycress kind of enters the equation. We started off this project and we selected 2 different crops that we were going to study in the laboratory. One, because it was an agriculturally relevant crop, and that's wheat. The other was Arabidopsis, because everybody and their dog in the plant world studies Arabidopsis. Our problem with Arabidopsis is it was very hard to get roots of Arabidopsis out and study them. And so we selected Pennycress because it's a bigger seed, gives us a bigger root, it was a little bit easier to work with. And so in the process of all that, we realized that we could do some nice things with seedlings with Pennycress that we could use to then kind of tease away what genes might be important to actually growing on the root exudates that the Pennycress provides. So this was an experiment that my collaborator Neil Olszewski kind of came up with. Instead of trying to work and grow the plants in soils or something like that, this is a very simple experiment we can do in the course of a week with seedlings, where we can really start to tease away what genes are essential for just growing with the root exudates. Because again, the nice thing about my algal system that we use is that it produces 2 carbon compounds that we know of, and that is maltose and glucose. But root exudates produce a lot of, or plants produce a much bigger variety of different compounds. And so this was a nice way to kind of get into that. And this is some of the approaches that we used for studying this. Really want to understand this interaction, and especially the genes that seem to be essential to establishing that. So what we do is we generate transposon libraries where we get 100,000 plus mutants that we can then sequence and determine what they are. We will mix that together with sterilized Pennycress seeds, put these in a flask together, germinate the seeds, and then we actually germinate the seeds for a couple of days before we actually add the library. Then we grow the library together with the seeds for about 4 or 5 days. And then we'll try to re-isolate the library from that and look at the library and see what the dynamics of the library, how the dynamics have changed over the course of that growth of plants. And that gives us an idea of which genes might be essential or beneficial, which ones are potentially not as important in this section.
How do you generate the library, the genes?
So it uses what's called a transposon. It's kind of like a bacterial virus. So that is the transposon goes in and randomly inserts itself in the genome, and it only does it once. That's really nice. So every 100,000, all those 100,000 mutants that we have, it's one mutation that you can actually go in and then sequence and determine what that is. So when we get sequencing, we get 100 million reads. We can then put those on 50,000 different mutants and determine what the population is at the beginning, and then what the population is at the end. If your mutant goes away, that was a gene you probably didn't want to mess with because that was essential. If your mutant improves, that might be a gene you want to mess with and get rid of because it might give you benefits. So there's certain tricks that come out of this experiment. It's fairly complex, but actually fairly simple once you actually get the final data. So putting out the experiment together, running it, this is a really nice way to establish it. And the results from it are actually quite telling. So this is what we end up with as results if you look at it from a bird's eye view. There are, I think, 3,200 genes in Gluconoacidobacter, and we were able to get reasonable data for about 2,300 of them. So about 2/3 of the genome we're able to look at. And the proof of concept here is this line right here. If the fraction remaining at the end is similar to the fraction that you started with, then all these genes here around 1 were not very important. And that's fine. That gives us some information, at least not under these conditions. The ones that start going away, this is where all the details are. These are the genes that are actually important, forming this association. And then the ones that increased might be worthwhile to kind of think about in the future as far as trying to do some improvement. And so nice proof of concept, nice reasonable gene coverage across the whole genome. We've got this manuscript submitted to Applied Environmental Microbiology right now to get a better understanding. This is what the data looks like. To do these experiments, we have to do quite a few replicates. So we run about 8 replicates when we do this to get our averages. And what you're seeing here is our cutoff in this case is all the genes that are below about 0.5. So we go through, we can pick apart kind of what some of those are, and then we can actually correlate that with specific genes and what they're related to. These are all the ones that are related to either sugars, pyruvate, or the citric acid cycle. And some of these we expect to be essential. There are genes that we don't see in this data here because you cannot get rid of them. If you knock those genes out, you can't even grow the organism on plants. So those are just the essential genes that are essential to the organism no matter what. These that we're looking here are the genes that are essential to growing with those root exudates. So it actually is pretty nice at explaining what's going on in the system. And then once we identify certain genes, we've gone back now and made a single mutation of that strain. And then we have ways to kind of test that against wild type to see if that really holds true when you just put a wild type strain side by side with one of these deletions to determine if they really are essential. And these 2 in red, we've made those mutants, and we've shown that, yes, if we now put these together and then try to grow them with the root or the pennycress in these flask studies, they do not grow very well. So that's kind of the proof of concept. On the opposite side, this is all of the genes that we found that actually led to slight improvements. Again, now here our numbers 1 down here, but we have some where we're getting a 4-fold improvement in growth. And we've done the same thing here. We start looking at what these genes are, what they might be doing. In some cases, we find a whole cluster of genes. So this is Gluconoacidobacter's gene 28, 53, 54, 55, and 56, just a cluster of genes in the organism. And if you knock out any one of those, we actually see an improvement in the growth on these root exudates. So all these things give us some nice targets of things to study. The problem with the experiment is that this is really only getting at the organisms potentially that live and grow in either the rhizosphere or are on the that are epiphytes. This growing these plants and flasks is not conducive to forming lateral roots, which are very important for endophytes. So this experiment's not the best experiment for getting to the heart of the matter, which is really want to know is how do we get these microbes into the root where we know that any nitrogen we produce is going to go right to the plant and not get wasted. So a lot of the reason why we focus on what class of organisms we're dealing with is we want to make sure that not only are we taking those root exudates, converting that carbon into nitrogen, but the nitrogen's getting to the plant. And so, again, we're still trying to understand the mechanism that govern that successful plant microbe interaction. This is the system that we use for that. So these are ragdoll setup that Neil came up with. They're really nice and conducive to forming lateral roots. So it's basically essentially the same experiment that a lot of kids do in elementary school, where you take a paper towel, put a seed inside of it, and let it grow. And you can actually watch the roots come out into the paper towel. The difference is our system is completely sterile. And then we add only one single microbe back to it. And when we're done with this, now we actually isolate and treat those roots with bleach to kind of kill off anything that's on the is an epiphyte. And we actually just try to isolate the endophytes as well as the epiphytes. So we actually do 2 experiments at the same time. We take a fraction and look at the endophytes, another fraction at the epiphytes. This data is still coming down the pipe. We do have some nice preliminary data on that as well. We don't have enough information to be absolutely confident this is working well. And that study, as I said, gets back to the endophytes. The other thing we did was we needed ways to actually trace our strains. So if we manipulate a strain in the laboratory and then we add it, we'd like to know that it's actually still there and contains the same manipulation when we're done. And so to do that, we actually went in and developed these tracer studies where we add a gene cluster for a compound called the oleucine. The oleucine is produced from tryptophan. It basically takes 2 tryptophans, puts them together. And when it does that, it turns purple. So you get this nice purplish, almost even black in some cases, phenotype that you can see with your colonies. And that makes it nice and easy to identify these on plates. So we've done this in multiple different organisms. One of these is Gluconacetabacter. One of these, I think, is Azotobacter. Another is Klebsiella. So we have this in quite a few of our model strains. And that's important because now when we challenge these, we can see. So if we put a mutation in and we say, is it really improving the growth on root exudates? We can put that together with another strain and we can track it. And it works out very well. It also works really well for if we go into the plants and put them in soil and then try to get the plant back at the end of the day and know for sure that the strain we put in is coming back. So it is a nice system and tool that we've developed along the way. So what have we learned and accomplished so far with this? We know that we can make extensive modifications to Gluconacetabacter and do quite a few different things, and we can make fairly complex strains from that. We have demonstrated that we can generate a very high ammonium accumulation in liquid culture and with algae. And we know that Gluconacetabacter will grow with Pennycress root exudates. So we get really good growth with that. We can get 7 or so doublings oftentimes. So it's doing just fine on root exudates. We can get Gluconacetabacter into plant roots in both wheat and in pennycress. But I'm going to tell you, most of the stuff we've done so far is with wheat. Wheat's just easier to treat the roots afterwards. It's a much bigger plant. You get a lot more biomass. And for these studies to work, we've got to get quite a bit of root back to be able to do endophyte studies. So most of our knowledge is coming from that.
Just a curiosity, how are you adding genes and gene clusters? Are you using CRISPR or another platform to add?
No, we're old school. So most everything we do is traditional cloning, adding antibiotics. If we go back to some of the techniques that we did for this, we'll drop an antibiotic in and then we remove it at the end. We actually have counter selection techniques. So we use sucrose toxicity with a gene that we put in, then spits everything out. So we can go anywhere in the genome, make any manipulation we want. When we're done, we leave nothing behind except the manipulation we want. So if I want to knock a gene out, I take that gene out and it's gone. And that's the strain I have, and that's the strain I move forward with. If I want to put a foreign gene in, I can put the foreign gene in with an antibiotic initially, and then I can take the antibiotic out. So I can put a foreign gene in. If I just want to manipulate, in some of the strains we made here, we actually took the regulatory system out for nitrogen fixation, so it's just always on. That stability of that might be a question. I'll kind of get to that later, but it worked, and it does produce nitrogen, and we'll get some of those. So we don't usually use CRISPR in this strain. I keep having people kind of nudge me to start working with CRISPR. My problem is that it already works for me, and developing CRISPR might take me 2 years. So if I have something that works, I kind of stick with it. Okay. All right, so we've done all those things. Our next step was, all right, if this is really seems to be working, we can get it into, we know we can get it into some plants, and at least in our laboratory system. We wanted to see if we could actually do this in soil. And I am going to point out that we realized that we were jumping over some major potential obstacles here, because we really don't know exactly how to get this into plant in soil. That was a big step. Everything we've done in the laboratory, we do in these systems where soil's not involved. So we don't have these dynamics between potentially the soil and the micro. We're really just dealing only with the plant in the micro. So we knew we were kind of skipping over that. But that was the plan of the study. That's what we wanted to go through and do. And so we started working with these containers and soils. We started this project a little over a year ago. And part of it was figuring out how to get these to grow well, and also figuring out what the media needed to look like to actually see a growth related curve with the nitrogen, where we can see a growth response curve with that. And we've established that using both peat for both pennycress and wheat. We did both at the time because early on with pennycress, we were running into issues where we wouldn't always see our colonies forming, or our endophytes forming with the pennycress. So that was one of the other problems we ran into. And then we also developed some different soil mixtures that include sand, peat, vermiculite. Most of this was done by Neil over in the growth chambers in the greenhouse, try to get some of this established. And so big question from it was, did we see a growth improvement with our enhanced nitrogen accumulating on Acetobacter diathrophic strains? And simple answer is not yet. We have run into quite a few issues where from one experiment to the other, we just don't get much of the Acetobacter into the roots when we go to soil. So this has become more of a hurdle than we expected, which meant we've kind of refocused our efforts here on trying to determine what the best methods are to establish these strain associations in plants when soil's in the mix, because that has become much more of a hurdle than we expected. So based on all this, we have a lot of questions of why we're running into these problems. One of those is, do our strains survive in soil? We did quite a few different experiments over the course of the last year with this. So we also need to know what's the best method to try to introduce. Should we be shaking these in seeds? Should we be trying to put dried cells on seeds? Should we be trying to add some kind of protectant to the microbes? And there's vast literature on this where people have worked for this, and a lot of people who figure this out, they then go start companies because you figure it out for one organism. The problem is that doesn't expand for Thylobacterium. What you learn there doesn't necessarily correlate to what you might need to do for Gluconacetobacter and Azotobacter. We know there's people that sell these inoculums across the world. We also know from a lot of people that study them that a lot of those inoculums that people are buying are not what people say they are. So you go in and you buy the stuff and you test it in the laboratory and you find out the strain that it says you have on the bottle is not what you actually are. You're growing some kind of yeast or fungus. So all those things we know are problems out there in the field. We also realized that we're going to have a major issue. In the laboratory, we've cheated in every possible way. We put only our microbe together in a sterile system, and we are never going to be working in a sterile system. So one of the big things we've realized is we have to understand how do these strains compete with the native strains that constitute this plant microbiome. And are we getting a sufficient number of these endophytes or epiphytes to provide a sufficient amount of nitrogen in it? These are some big questions that we realize we have to refocus on. And that's where we've kind of shifted our attention. That's not to say we've given up on the soil stuff. We actually are set up now with some other grants that are coming in that allow us to continue to work on this, even once this money from Forever Green completely dries up. But that's where most of our focus is at the moment. And really kind of trying to dig into this idea of synthetic core microbiomes. This is that just like us, for like humans or any other model biological system, we're finding in a lot of plants that there's a small core of known microbes that are almost always present when you look at these. And there have been some nice research that's come out of different research groups that realize that while you have a lot of diversity what's in soils, most plants themselves seem to establish this core microbiome that is dominated by a much smaller number of class organisms. I won't say they're always the same organism, but you're always going to find some Pseudomonas. You're always going to find some Chrysciobacteria. So there's certain strains that are pretty predominant. And through a number of different various approaches, there's groups that have isolated a subset of these strains that form what they call a stable synthetic core microbiome. And that they know they can add and continually see it growing and working with their plant roots. And so we've been starting to try to determine how well our organisms are able to integrate into those. And that's where our core research is at the moment. So does Gluconacetabacter integrate into and propagate within a synthetic core microbiome? We isolated and identified some, or obtained a synthetic core microbiome. And then we looked to see if it'll actually integrate into that. And the answer is that yes, it does. We actually do see an increase. Now, we're not seeing the types of numbers that we were seeing before, but we're also not seeing these synthetic core microbiomes wiping out our strain. So it's able to integrate in and survive to some degree.
In what conditions is this?
So some of this is done in flasks. Some of this is done in the I forget the ragdoll setups. We have not done soil yet, but people have worked with these in soil as well. So that would be a later step. That would require more funding. We are not funded at the moment to go into soil with that. But the other systems we have for testing it in the ragdolls and testing it in flasks, these are flask studies specifically, where we put all of them together in the flask and say, are these core microbiomes going to wipe out our strain? And is this going to be a dead end before we ever start? So that was one of the first things we wanted to do. And it's kind of a major proof of concept that for our NSF proposal, they wanted to see. That was what I think kept us from getting funding in the last round on that. And so we do have numbers for that. But we also tried another model endophyte that we did not select, and that's Klebsiella. This Klebsiella used to be called pneumonia. They've changed the name of it now. But it's a model that's been studied for a while. And that one not only survived, but it actually did pretty well. It actually integrated in the genome much better. So there's a difference in certain strains. And that's where we want to be very careful in this project to make sure we're not developing tunnel vision with any of our strains. We did a lot of work in Azotobacter. We went into plants and realized Azotobacter is not a very good model for plants. We did a lot of work with Gluconacetabacter now, and we do know we can do quite a bit with plants, but we don't know that we've picked the best strain. We just know that we have a strain that does some nice things in the laboratory. It's struggling a little bit more. And Klebsiella might be a better strain for integration. And so we might be going back to the drawing table on that. We actually have quite a bit of data for a number of these different strains. And even though some of these things don't work, we still work on them in the background. Like Azotobacter has benefits over all of these guys because it can do this aerobically. And so it could potentially survive in a part of the soil that others couldn't. Problem is, it doesn't grow very well with plants. So we're still in the background working on trying to get it to grow well with plants. We're still working on Gluconacetabacter. We're not going to give up on that. We have quite a bit of data that tells us how to maybe improve that. So we might be able to make improvements over time to it. But we're also looking at Klebsiella, and we also want to be aware that there might be others. And that's where we get into this other funding that's going to kind of help us do some of this work in the future. So kind of continue this. But it also opens up a new area for us, which is trying to understand what these microbiomes look like for some wild plants. So we are doing this through LCCMR. specifically going and looking at healthy native prairies. But we're also have a component of that that looks to see if we can't transfer some of those microbes over to agricultural crops. So it enables us to survey different plant microbiomes, also allows us to select several different crops to determine if native plant microbiomes can be established in those. And we've done so much work now with wheat, so much work with pennycress, I don't think we're going to abandon those anytime soon. So those will be part of it, but we're also looking at some other strains, some other systems where gluconacetabacter might be better. So sugar beets might be a much better selection for gluconacetabacter. So we're looking at some of those crops as well. The other thing we've done is we've been trying to dig in a little bit more and work some more with some of the folks that are doing some of these crop studies here locally. And so last fall and this spring, we've been working with some of the people that are growing both camelina and pennycress, as well as some winter wheat out in the fields. And we've been trying to just figure out, well, what are those microbiomes look like? And specifically, what are the endophytes? What are the endophytes and epiphytes that are in those look like? So we can figure out who our competition is. And based on those studies in the past year, we've actually isolated quite a few different microbes. And a lot of those are similar strains, so we're finding these core microbiomes. So that's a nice positive piece of that. But we're starting to get a little bit of an understanding and a baseline for what's happening in these things in the field right now, and trying to identify if there are other strains we should be thinking about integrating, because that's who's already there. So that's one of the things we're looking at doing there. What is the status of these soil studies and with pennycress and diazotroph and gluconacetabacter? We are still looking to understand, do our strains survive in soil? And that's something that we've not really got a solid answer for. So we've just got a lot of experiments looking at that. This method of introduction, these are the things we're still trying to understand as well. And then how do we improve the strains so that they compete with these other native strains that constitute? Because we do believe that with these other studies, similar to what we did with the TN-seq studies with transposons, we can identify certain genes that maybe help compete, or we can steal genes from other organisms, maybe help them. We usually like to try to do the first because we don't want to do too much genetic modification that makes them transgenic. Most of what I've shown you in this study is genes that we delete, not adding things to, but we're not afraid to add things to it either. We're not afraid of transgenics. So these are some of the big points that we are kind of focusing our attention on. And then the last thing I want to kind of finish up with is we do also have some nice recent victories. And that's related to one of these things that I didn't talk about at the beginning, but the strain stability is an issue. So most of our modifications we make, we try to produce lots and lots of ammonia. And the problem is that to do that, we deregulate the system so it does that. And that leads to strain instability. So we know that we can get these things to work in the laboratory for a few weeks. We also know that to make them work out in the field for an entire season or for several years, these probably aren't going to be long-term stable because they will eventually evolve. And to some degree, that's a benefit, but we'd also like to try to address that. And so while we have had some success both in azotobacter and gluconacetabacter for ammonia, it's important to get back to our very first few papers where our target was never ammonia. Our target was always urea. And there's a reason for that. And that's because urea is less toxic, it's less volatile, it's easier to transport, and we can turn it into a terminal product, meaning that almost every organism that uses urea breaks it down with an enzyme called urease. So if you simply wipe out urease, you wipe out the ability of the organism to break it back down. And for some of these organisms, gluconacetabacter and azotobacter, urea doesn't inhibit nitrogen fixation. So if you start producing lots and lots of ammonia, you start producing lots and lots of inhibition. You actually slow down the process of nitrogen fixation. And with urea, our strains are blind to it. You can be bathing in urea. They'll still keep fixing nitrogen if they think they need nitrogen to sell and can't utilize the urea. So for about a decade, we've been working on this with very little improvement. But we about a year or two ago, kind of refocused our efforts on trying to get urea numbers back up. And we developed this strategy where we went into some of the systems that people use right now to produce a lot of your amino acids. The issue with Azotobacter when we were doing this, and most of this is proof of concept is done in Azotobacter. Azotobacter doesn't have the enzyme that makes urea. It does have the enzyme that breaks it down. We wiped that out, and we do get a little bit of urea, but never high concentrations of it. And so we've been trying to get a full urea cycle in Azotobacter for a decade. We recently got a little bit of success on that, but then we kind of plateaued and hit a limit. So we got about a 10-fold improvement for what we were getting before that. But then we went back and deregulated this system by putting a gene in that overcomes this feedback inhibition for arginase. Arginase is the last metabolite you have before you break off urea from arginine and form ornithine to kind of recreate the cycle. It's what you and I use to get rid of urea from our systems and excess nitrogen. So using this approach where we made 3 different modifications to Azotobacter, we've been able to actually get dramatic improvements in urea. The reason why this is fairly beneficial again is Azotobacter doesn't see a urea. So this becomes terminal. The other thing that's really nice about it is we don't see a growth defect. The Azotobacter is making a little bit of urea. It's kind of just bleeding off from the system. We aren't actually messing with nitrogen fixation at all. We leave nitrogen fixation as it is. This is a side product, and we're just leaking a little bit of urea out. And that urea can't get reconsumed, so it can't really find a way to evolve and take the urea back without stealing an urease from other organs. That's a pretty big evolutionary step to get another enzyme. And so this kind of shows, this is where we were at about 10 years ago. This is where we were at about 2.5 years ago. And this is where we're at today. We're producing about 6 millimole of urea. We produce about 25 of ammonia. The benefit of urea is there's 2 nitrogens for every molecule. So that's like getting 12 millimole of ammonia. And what this looks like if you do these head-to-head with algae, what we have here is our wild type strain growing together with our algae. We don't see hardly any algae growth. This is our ammonia producer when we've got it all amped up and producing as much ammonia as possible. And this is our urea producer. And you can see we're getting pretty similar yields, pretty good growth. This is going to be a much more stable genetic system. So we actually have solved one of our longer term goals now earlier, while we're still struggling a little bit with some of our short term. So on the horizon, we're still learning a lot about how our strains integrate into core plant microbiomes. We're seeking additional funding to give this an obstacle that we know we've got to overcome at some point, more attention. The LCCMR project we just got funded is going to provide sufficient funding to dig into this hurdle that we will need to overcome to some degree. It doesn't really allow us to do the type of really extensive experiments to try to do strain modification, but it does at least allow us to see what else is out there and potentially study some of the strains that we're introducing and how we would integrate our strains into that. We have a number of target genes based on these studies with epiphytes and endophytes that we've identified that we would like to kind of add to our strain because we think it might help them grow better with our root exudates, and also because we think it might help them grow better as an endophyte. We've identified some other key microbes that are really good at growing either as an epiphyte or growing in the rhizosphere, or that are really good at establishing themselves as endophytes, significantly better than our Gluconoacetobacter model that we initially selected. And we do need to give more attention to the methods of strain introduction. We know that's going to be something we have to overcome. And that might be something where one day we just try something and it works really well and we're back in business. And maybe something where we have to do some strain modification, get a strain that not only makes everything we want it to make, but is also really good at surviving that initial time that it needs to be stored with the seed or the application to go on this. So those are some of the things that are on the horizon. Quite a few people have been working on this project over the years. Ben did a lot of our early work. with the Acinetobacter to turn it into the same type of strain that Azotobacter is with excessive nitrogen production. Quite a few other folks, Neil, of course, working with us, doing a lot of the plant growth itself. A lot of undergrads, we had an army of undergrads, especially on the Forever Green, because it was a seed grant. Wasn't really quite enough to fund a graduate student. So most of our people that have worked on this have been under Neil's and my supervision and under the supervision of other graduate students doing this. So quite a few undergrads have worked on this project. And then we've got Aaron and Kobe, who are newer grad students that are really kind of picking up the torch and continuing on some of the work with the plant stuff. I was happy to take questions all along, but I'm happy to answer any other questions before I'm done too.
All right, thanks, Brett.
You identified 20-some endophytes naturally occurring in Pennycress.
Yeah.
Were any of those among those 6 or 7 that you talked about?
The 7 core includes a lot of Pseudomonas. Well, the core includes the core that we work with in the laboratory for our model includes a Pseudomonas strain. It includes Chrysiobacterium. I don't even know if I'm saying that one right. It's a nice golden colored microbe. You always know when you have it. It includes an Enterobacter, and then includes a couple other strains that have been renamed. Those are all members of classes. So Pseudomonas is a very well-established class, and we've actually found quite a bit of Pseudomonas. It's not the exact same Pseudomonas strain, and some of those might actually be they're endophytes, but I can't tell you for sure if they're good endophytes or bad endophytes, because some of these endophytes are growth promoting, and some of them are pathogens. So we have not studied, like after we isolate them, whether they are pathogens or not. And so yes, the answer to that is some of those 4 organisms that we're going to have to contend with are being found. Quite surprising how much Chrysiobacterium is there. In fact, we thought about maybe trying to go in with another seed grant. We had nothing related to it. Chrysiobacterium, a lot of them are being studied right now because they're anti-nematode. And so we're interested in that just because we've been listening to quite a few people talking about the nematodes issues and some of that. Okay, this might be a way to think about taking some of those and looking at that. I even talked to one person after one of the meetings last month, who's doing some of this work, I think, down at Waseca, and kind of mentioned to her, yeah, we might have some strains just to see what it would even take to test some of those. We're not just looking at nitrogen fixation. We also scanned some of these strains for phosphate solubilization, potassium solubilization. One of the other ones we're really interested in is silicate solubilization. It turns out that one way to fight off a lot of different pathogens for plants is to make silicate more bioavailable. And you would think plants are bathing in silicate, so they should be really good at getting silicate out. But you can put silicate in certain forms and actually improve dramatically how plants fight. So we're actually interested in that because there are microbes that I don't know that they are specifically trying to solubilize silicate. They're trying to actually keep carbonate in the right form. And the enzyme, I think, is just kind of does the silicate solubilization. So we're interested in all those things as well. Yes, the short answer is we do find some of the same 4 microbiome strains there. And we do from time to time, especially for Enterobacter, that's a very common organism for an endophyte. So we do find strains that are in that same class. They aren't necessarily the exact same strain. And our strains that we're working with, some of them are related. So Azotobacter, for example, is a very close relative of Pseudomonas. However, you put Pseudomonas with a plant and it does beautifully. You put Azotobacter in with a plant and it does horribly. The problem is Pseudomonas, most of them don't fix nitrogen, and Azotobacter does it beautifully. So if I could somehow cross the two, it'd be off to the races. And that's one of the things we are looking at in the background is as we do all these, these core microbiomes, we're trying to figure out how to make azotobacter quantum Which is why I say we haven't given up on so.
You know, when you made those isolations of those 27 endophytes, were they from one variety or line of Pennycress and one of Molina, or was it from like a breed?
They were two different times we went out into the field. Every time we did that with David Marks. I can't tell you what exact strain they were. I'm sure we could find it out, but I think we tried to stick to the standard strain and not any new mutants. I know we think that we're using one of the older Pennycress, and Neil and I have been trying to decide if we should switch to some of your newer strains there, because all of our data is established with the older strain, and we're afraid that maybe we're going to see some slight variances if we go with new strains. Same thing for wheat. I think you guys are using a very different strain of wheat than our strain of wheat we've been working with. We're actually growing it ourselves right now, so we're working with the same strain until we get the studies done.
What's the name of your wheat?
Give me the name and I'll tell you.
When did you start working with it?
5 years ago.
Laker,
No,
Barney, Rotsey.
So it's a winter, right?
No.
Or was that just a field that you?
the winter wheat's what we get in the field. This is not what we're. This is just the standard. Neil would be the right person to answer the question.
But I'd be interesting to know if there's
Bob
variety.
something.
Bob White.
Bob White.
It's from Mexico. It's adapted here.
And again, we were using this for just to do the laboratory studies. We may at some point transition to something that's more relevant to something that we know is going to be more useful in Minnesota. We were doing that more than anything so we could isolate endophytes. So we started off with Bob White. I think that's what we have.
Yeah, that's not good. It'd be interesting to know, just as a plant breeder, are different varieties better or worse hosts for some of these?
Yeah, love to do it.
We're probably selecting for ability to host the beneficial ones or check the.
I think once we get past some of these initial hurdles of understanding how we establish, we would love to look at other breeds. We'd love to look at a lot of other plants. I'm still not convinced that Acetobacter, as is right now, won't work beautifully in another crop. There's just not a lot of people out here growing sugar. So that's where most people do their grow. There are some sugar beet growers. We're interested in trying to play around with sugar beets. The other thing about Acetobacter is our hurdle is getting it in the plant. But in Brazil, and I'm not a plant breeder, but I think most of it's cuttings. So you're transferring probably the Acetobacter with the plant once you get it in there. So if you get it established, it may just be transferring it over already. So we don't know for sure. So we're actually kind of interested in the perennials for that reason. It's maybe able to get it established over the course of several years, and maybe it evolves on its own to become better.
If you look at elderberry or hazelnuts, those are the cutting propagated crops that we work with.
Yeah. And I don't know that's going to be a big change from Pennycress and wheat. So we picked wheat and Pennycress initially, or we picked wheat and Arabidopsis originally because there were several papers that said very specifically people had gotten it and had pictures of the Acetobacter in the root. So it's like, we know they're going to be endophytes in here. That's why we started off. We don't do a lot of microscopy ourselves. We don't really want to do a lot of microscopy ourselves. The bleach works out just fine for us for identifying.
Question is about the LCCMR project. So you're going to survey native prairie microbiomes. And is the hypothesis that if you could establish those microbiomes in crop fields, you get a better set of ecosystem services from that microbiome than the one that's currently found in a crop field, or is it?
The hypothesis is that most people who have been studying agricultural crops have been studying agricultural crops in agricultural fields. And because of that, over the course of, especially with people that, keep the same crop in the same field year after year after year, they're probably perturbing microbiome dramatically. So our idea there was, let's try to find native prairies that are less perturbed. The problem there is that when we start talking to all the people, the native prairies, a lot of these are native prairies now, but it's only like in the last 20 years, they've kind of reverted them back. You find some that are undisturbed. And it's studied. There are some people that have done some work with different plants for those. But yeah, we just want to make sure we're not missing out on certain things. If you go back into the really, really old literature where people were looking at the nitrogen fixers, there's a number of different organisms that nobody works with it. But it's like at some point they did. And they probably did because they were finding it in some of their crops. And then somebody starts planting crop for corn year after year after year before long, your soil microbiome shifted on. So that's part of the reason why we're going in there. The other reason is most of the microbiome studies just look at the soil. And we want to look at the endophytes and the epiphytes and the soil all together and look at who's really in close proximity. Proximity for us is key, right? We can make all this nitrogen out in the rhizosphere. How much of that actually gets to the plant? And even people that supply fertilizer tell you that maybe 30% the nitrogen actually gets into the plant. A lot of crops. And so we want those associations really close so that all the root exudate or even the internal metabolites, if they're inside the root and they spit out nitrogen out to their surroundings, they're inside the root already. The plant should have the first crack at that nitrogen. So that's part of it, is we're trying to do some studies in a way that other people haven't done those, which we haven't explained to LCCMR because we can't put that in an LCCMR proposal. But that's our predominant reason for trying to get into that. It's not to reinvent the wheel. There's plenty of people doing microbiomes in prairies. It's kind of the way we're doing it and the selection of prairies that we're doing, because we think some of these might be really good for finding things that deal with drought in the future. So if you go into a prairie that typically is a sandy prairie where you deal with a lot of issues with hydration or keeping your hydration forward, that might be a nice place to look for drought tolerant microbes that might be beneficial as well. So we're trying to keep all those on the horizon, establish that baseline, and get a nice core of several hundred strains that we can say, are these culturable in the laboratory? Would they be amenable to work? And are there other nitrogen fixes that we should be working on because they're just naturally present there anyway? And again, if I was working in Brazil right now, I'd probably be fairly well set, be doing a lot of experiments. But because we're trying to do stuff with the Midwest, we want to kind of understand what's going on in the Midwest, the Midwestern markets.
Cool. Any other questions?
See anything online?
Cool. All right. Thanks again, Brett.
Thank you.